Good morning and hello to everyone. Today we are from Group 2 are going to present about muscular system. The presenter is Sabrina, Maisarah, Aqif and myself Izzati. So I hope that all of you can pay full attention to our presentation today. First of all, we should know what is muscle. So what the first thing come to your mind when we talk about muscle? Basically, muscle are composed of contractile tissue. Our body have a lot of muscle that help us to move our body. Muscle are capable of changing its length by contracting and relaxing. Most animals rely on muscle for movement, and we should know that only a few animals are non-motile or known as sessile. Most of non-motile animals live in aquatic environment. There are three types of muscle tissue, which is smooth muscle, cardiac muscle, and skeletal muscle. This is an additional information. Over 650 skeletal muscle make up approximately 40% of the weight of an average human. Thus, muscle tissue is 15% more dense than fat tissue. You can refer to the diagram in the slide, which is anterior view of some of the major superficial skeletal muscle. So next is microscopic anatomy and physiology. We will see the details about skeletal muscle. Skeletal muscle is attached to the skeleton by bands of fibrous connective tissue called tendons. Skeletal muscle was arranged in antagonistic pairs. Antagonistic muscle only can pull and cannot push. When one muscle of a pair flexes the joint and bends the limb, its antagonist extends the joint and straightens the limbs. Example for antagonistic pair is bicep and tricep. Basically, when bicep is contracted, the tricep will relax, and vice versa. If the bicep is relaxed, then the tricep will contract it.
Okay, Assalamualaikum and hi everyone. My name is Sabrina, and now I will proceed the presentation with the microscopic anatomy and physiology. So before this, we have learned about the function of muscle system in general. Now I will introduce the structure of muscle fiber and their function in order for our body to move well. So as you can see in this slide, we are required to have these 4 skeletal muscle fiber, and the first skeletal muscle fiber is sarcolemma. So sarcolemma, or which act as plasma membrane which separate the outer environment from the muscle cell, and the sarcolemma will form a transverse system, also as T system. In this T system, the structure we call as T tubules, and these T tubules will deep down to come in contact with the sarcoplasmic reticulum. So we move to the second skeletal muscle fiber, which is a sarcoplasmic reticulum. So this sarcoplasmic reticulum consists of expanded portions of modified endoplasmic reticulum. So this modified endoplasmic reticulum will serve as storage sites for calcium ions, which are essential for muscle contraction. And in this structure, or in this sarcoplasmic reticulum, have the hundreds or even thousands of myofibrils, which are the contractile portions of a muscle fiber. So we move to the third skeletal muscle fiber structure, which called as myofibrils. So the structure of myofibrils are cylindrical and run the length of the muscle fiber. When the myofibril contract, it has a contractile structure in the sarcoplasm and have the light and dark bands, or we call as the striations. These bands are responsible for skeletal muscle striated appearance. The electron microscopy reveals that the striation of myofibril are formed by the placement of protein filaments within contractile units called sarcomeres. So what is sarcomeres? Okay, sarcomeres is one of the skeletal muscle fiber structure which act as the unit of contraction with myofibrils. And sarcomeres consists of two types of protein filaments, which are thick filaments and thin filaments. The thick filament made of myosin, and the thin filament made up of actin. And as you can see in this diagram, which show the Z line, is the actin filament. So the I band is light color like this one in this diagram. It contains only actin filament attached to Z line. Z line is this one, as you can see in this diagram. And the dark regions of the A band contains overlapping actin and myosin filaments, and its H zone has only myosin filaments. In this slide, we can conclude that the skeletal muscle fiber structure, like first from the muscle fiber, from the tiniest cell in this structure to the myofibrils, and move to the sarcomeres, which is thousands of fibril will make sarcomeres, and the sarcomeres will contract, and the contraction of muscle will occur. Okay, now I will explain about the sliding filament model. So as you first I will explain about the structure of the sarcomere and the protein filament, or we can say as the actin filament, which is we have myosin and actin. So first of all, as you can see in this diagram shown in this slide, first of all, I will explain about the structure of this model. This model is the structure of sarcomere and the position of the myosin and actin. So firstly, the actin filaments are found at both ends of the sarcomere. One end of each filament is attached to a Z line at one end of the sarcomere. As you can see here, the actin filament, which is this at the end of the sarcomere, which is this the whole diagram. This whole diagram is a sarcomere, and the small part at the end of the sarcomere we have actin, and it attached to Z line, which is we can see it like branches, and the other end is suspended in the sarcoplasm. So sarcoplasm, which act as medium, such as the same function like cytoplasm, like that. So the second one is myosin filaments are suspended between Z lines, which is the orange structure. So this is the condition when the sarcomere are relaxed, and the second one, the second diagram is the condition when the sarcomere are contracted. So we move to the next slide. Okay, now in this slide I will explain more details about the mechanism of this model, how it works. So firstly, when the myofibrils of muscles fiber is contract, the sarcomere will shorten, and when it shorten, and in the inside of the sarcomere, the actin filaments like pass through the myosin filament and approach each other. This cause the I band, which is this one, I band to shorten and H zone nearly disappear. And this is the condition when the sarcomere relax. You can see the H band, or we can say as H zone, we have the distance, and when it contract, the H band disappear. We cannot see the H band. So that's the simple mechanism of the sliding filament model. I will pass to the next presenter.
Now we will continue with muscle contraction. This is one of the important part in our topic. To have a better understanding, let's watch a short video.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
So, when a muscle fiber contracts, actin filaments slide past the myosin filaments and approach one another. Then, you can see the diagram in my slide. It will cause the Z lines to move toward each other. The sarcomeres will be shortened and the filaments remain the same length. Working muscles require ATP. Myosin filaments break down ATP and form cross bridge that pull actin filaments toward the center of the sarcomere. How does it happen? The myosin filaments attach to the actin, forming a cross bridge. The breakdown of ATP release energy which enables the myosin to pull the actin filaments inward, contracting the muscle. When ATP molecule binds to the myosin head, the myosin detach from the actin and the cross bridge is broken. When ATP is broken, the myosin head can attach back to an actin binding site further along the actin filament, and the process is repeated. Let's see a short video to make sure all of you understand.
And myosin. When a nerve impulse arrives at the muscle, it causes a release of a chemical called acetylcholine. The presence of acetylcholine causes depolarization, enabling calcium to be released from the sarcoplasmic reticulum. The calcium binds to troponin, changing its shape, and so moving tropomyosin from the active site of the actin. The myosin filaments can now attach to the actin, forming a cross bridge. The breakdown of ATP releases energy, which enables the myosin to pull the actin filaments inwards, contracting the muscle. This occurs along the entire length of every myofibril in the muscle cell. When an ATP molecule binds to the myosin head, the myosin detaches from the actin and the cross bridge is broken. When the ATP is then broken down, the myosin head can again attach to an actin binding site further along the actin filament and repeat the process. This repeated pulling of the actin over the myosin is often known as the ratchet mechanism. This process of muscular contraction can last for as long as there are adequate ATP and Ca+ stores. Once the
Next, we will discuss the use of ATP in contraction. Remember, cellular respiration does not immediately supply all of the ATP that is needed to the cell. Muscle fibers rely on creatine phosphate. This is the chemical reaction of creatine phosphate and ADP. When creatine phosphate is depleted and the cell still needs more ATP for muscle contraction to occur, it undergoes fermentation and will supply ATP for a short time. The lactate will build up and the fatigue will be produced. The continued intake of oxygen will restore cells to the original state, so it will offset oxygen debt.
Now, before we end our presentation, we've got two more concepts to cover. We've got muscle innervation and also the role of calcium 2+ ions in muscle contraction. So let us begin with muscle innervation. To start off, let us watch this short video so you can have a better understanding of the topic beforehand.
The connection between a motor neuron and a muscle fiber is called a neuromuscular junction, which is basically a chemical synapse between the nerve terminal and a specialized area of muscle cell membrane called the motor end plate. When an action potential reaches the nerve terminal, it causes the release of the neurotransmitter acetylcholine into the synaptic space. Acetylcholine then binds to nicotinic receptors on the end plate. Nicotinic receptors are ligand-gated ion channels. Upon binding to acetylcholine, they open to allow sodium to enter the cells, depolarizing the cell membrane, producing the so-called end plate potential. An action potential is generated in the muscle cell only when the end plate potential reaches the threshold required to activate voltage-gated sodium channels located outside the end plate in the neighboring membrane. When activated, these channels allow faster influx of sodium, further depolarizing and eventually reversing the polarity of the cell membrane. At this point, voltage-gated potassium channels open for potassium to move out, quickly returning membrane voltage to its original resting value. Once generated, the action potential spreads like a wave, thanks to similar voltage-gated ion channels located throughout the muscle fiber. The action potential also runs deep into the fiber via T tubules, to reach the sarcoplasmic reticulum. Here it activates voltage-gated calcium channels, releasing calcium from the sarcoplasmic reticulum into the cytosol of muscle cells. Calcium then sets off muscle contraction by the sliding filament mechanism. This mechanism is described in another video. Another important component of the neuromuscular junction is the enzyme acetylcholinesterase. This enzyme removes all acetylcholine molecules that do not immediately bind with the receptor, and those that are done activating a receptor. The enzyme action essentially terminates synaptic activation, giving the muscle time to relax, and thus preventing continuous contraction that would result in muscle spasms.
Muscles are stimulated by motor nerve fibers. Now, motor nerve fibers have many branches, but they all end in axon terminals. These axon terminals are in close proximity to the sarcolemma of muscle fibers, but they are separated by a synaptic cleft. This region is what we call neuromuscular junction. Now, let's look into the term of neuromuscular junction. Junction means a place where two things meet. Neuro means neuron, muscular means muscle. So when we put them together, they literally mean where neuron and muscles meet. So let's look into the structure of axon terminal. In the axon terminal, we have synaptic vesicles that contain neurotransmitters that we call acetylcholine. Now, let's look into how nerve impulses are transmitted from our nerve fibers to our muscles. First of all, nerve impulses travel down our nerve fibers and reach the axon terminal. Once it reaches the axon terminal, it stimulates the synaptic vesicles to release acetylcholine into the synaptic cleft. The acetylcholine then diffuses quickly and binds the receptors of the sarcolemma. Now, impulses spread all over the sarcolemma, down to the T-tubules, and then lastly reaches the sarcoplasmic reticulum. Now, this stimulates the sarcoplasmic reticulum to release calcium 2+ ions, and this calcium 2+ ions causes the contraction of sarcomeres, which is actually causes the sliding of the filaments, and this in retrospect causes the contraction of our muscles. Now let's look at the individual components of muscle contraction in more detail. First, let's look at actin filaments. What are actin filaments? Actin filaments slide past myosin and causes contraction. Calcium 2+ ions are needed for myosin to bind to actin, and myosin filaments pull actin filaments by means of cross bridges. Now, myosin filaments are azymatic and they actually split ATP into ADP and P. ATP is used to supply energy for muscle contraction. Let us talk about two proteins that are associated with actin filaments. First, tropomyosin. Tropomyosin are threads that wind around an actin filament, while troponin are placed at intervals along the threads. And troponins are the proteins that will bind with calcium 2+ ion. Now let's look into how calcium 2+ ions actually cause muscle contraction. So first, calcium 2+ ions from the sarcoplasmic reticulum combine with troponin, and this actually forms a troponin calcium 2+ complex that pulls on the tropomyosin and shifts their position. And so this actually exposes the myosin binding sites. Now let's look into the first step. So firstly, we must know that the myosin filament is actually composed of two globular heads. And so the heads actually function as ATPase enzymes that split ATP into ADP and P, which still bind to the myosin heads. So first, what happens is that ATP will bind to the myosin head and hydrolyze, and so they will form ADP and P. And this whole process actually activates the heads. Now, the heads are free to bind to the actin filaments, and so as they bind to the actin filaments, they form a cross bridge. And as this cross bridge forms, the ADP and the P are released from the myosin head. And this actually causes the myosin heads to bend back to the original position and pulls the actin, causing what we call a power stroke. Now, power stroke pulls thin filaments into the middle of the sarcomere, which takes us to the last step, which is the cross bridges are broken when the heads detach from the actin, and so this breaks the cross bridges, and then ATP is free again to bind to the myosin heads, and so the cycle begins again. And that, my ladies and gentlemen, is how we will end our presentation. We hope you learned something in this presentation, and if you ever need to, please come back again to learn more on this topic.